So now we're going to move into our next part of the agenda, and I want to go ahead and turn that over to Bob Ellis, who will introduce our next speaker.
Okay, thank you. It's my pleasure and privilege to introduce a very valued colleague, Dr. Richard Bowen, as our next speaker. And Dick's going to speak about the science behind COVID-19 and SARS CoV-2. Dick is really well qualified to talk about that because over the years he has conducted a lot of coronavirus research, SARS-1, did research with that in animal models, and one of the really significant things that he has done his research over the years was put MERS back into camels, actually, in Colorado, both dromedary and Bactrian camels, and established the route of infection, what happened after infection, and the shedding, high amount of shedding of that virus from the animals that Dick had in his research. I used Dick and his research and his overall laboratory research a lot in training sessions that I do because he is the epitome of versatility and variability. He has a list of animal species that goes forever as far as the ones that he has used in animal modeling of human diseases and animal diseases, and definitely, probably most importantly, zoonotic diseases, those that come from animals to humans. The Dick got his DVM, earned his DVM at Colorado State University and his master's and PhD. He's currently a professor in the Department of Biomedical Sciences, which is within our College of Veterinary Medicine and Biomedical Sciences. And he also has a joint appointment in the Department of Microbiology, Immunology, and Pathology. And I want to point out that one of the significant awards, among many awards that he has had over the past, but one of the significant recent awards was that he was awarded the 2020 Lifetime Excellence in Research Award by the American Veterinary Medical Association. And this was primarily for his research with zoonotic diseases. So without further taking up his time, I'm going to turn it over to Dr. Bowen. Thank you very much, Dr. Bowen, for being on our symposium.
Bob, thanks very much. You're very kind. So I think you all, I don't need to give any of you the history of SARS-2 and COVID. We kind of jumped on this in March when we first got the virus, and our lab basically, as Bob kind of intimated, specializes in looking at host range for zoonotic pathogens and pathogenesis, and then we also do a lot of work on countermeasure evaluation. And so I had a call from a reporter in Hong Kong early on about when they first diagnosed a dog with as being positive for SARS-2, and they were throwing dogs out on the street in Hong Kong, and I figured we ought to jump on the dog and cat situation because it looked like a fairly substantive problem. And so I'm just going to walk you through a number of studies that we've done to define host range and develop animal models for SARS-2 infection. So let me see. There we go. So as I just kind of indicated, there was a report of a PCR positive dog in Hong Kong. I don't remember exactly when it was, that caused a great deal of panic. And subsequently, there have been a few other positive dogs and cats. There was a tiger diagnosed with SARS-2 infection, and additional cases in cats. And there was a relatively strong public response to that. People very anxious about what role these animals might play in the whole COVID epidemiology. And so our approach to experimental infections with both dogs and cats was to do intranasal inoculation of the virus at a reasonable dose, not a huge dose. We're basically using 10 to the 5th plaque-forming units. You can see the kind of setup that we use. I really like to do experiments in animals that are kind of free-ranging within our biosafety level 3 rooms, and that's what we did with the cats and the dogs. Cats, I'm a big fan of cats, but I'm not a huge fan of cats doing experimental infections. They're kind of hard to handle, but these little cat bags worked pretty well. So we inoculated both species, and as I'll tell you, with cats, we did a substantial amount of work trying to understand whether they could transmit to roommates, basically. But the summary on the dogs, we only did 3 dogs because the work that we did and the work that a few other groups have done indicate that they're just really not very susceptible to infection. And so we infected 3 adult beagles intranasally, and then monitored them, kind of standard clinical monitoring, temperature, clinical signs, nasal discharge, all that kind of thing. And then did oropharyngeal and attempted nasal swabs on them over the first few days after infection. And so what we saw was that there was really 0 clinical disease. We didn't even see a hint of a fever in these animals. We were unable to detect virus shedding, but they did seroconvert, as you might expect, kind of a low level seroconversion. And as I indicated, this is similar to what other people have found with dogs, and so kind of the conclusion here is that they simply are not much of a risk factor as far as I'm concerned. Cats, on the other hand, we started off infecting a couple of cats with, again, intranasal inoculation of a bit over 5 logs of virus. The cats, similarly to the dogs, did not display any signs of clinical disease whatsoever, no fevers, no loss of body weight. But in contrast to dogs, they shed a lot of virus nasally and in oropharyngeal fluids for the first few days after infection. Moreover, we introduced uninfected cats into the inoculated cats the day after infection, and we readily got contact transmission, which in retrospect is not too surprising considering how much virus they were secreting. Cats developed just a whopping amount of neutralizing antibodies, so robust seroconversion. And I'll show you some pictures of the pathology. The kind of take-home message for pathology is that this is, we get some very mild lesions in the lung, but it's primarily an upper respiratory tract disease. And we also did radiology and didn't see any hint of pulmonary involvement radiologically. So this just shows you the amount of virus that's coming out of these cats, both nasally. The way we do this nasal shedding is similar to what we do with ferrets and influenza, where we restrain the animal. With ferrets, we have to anesthetize them. With cats, we really don't. And then we kind of just drip a little bit of a cell culture medium into their nostrils, and they sneeze, as you might expect. Then we collect that into a petri dish and assay it for virus. And so you can see a number of cats here, and you don't need to really scrutinize each individual line, but what you can see is that the peak viral shedding is on day 2 or 3 post-infection, and it's substantial. It's, in this particular case, it's like 10 to the 4th, 10 to the 5th plaque-forming units per milliliter. The oropharyngeal swabs, a very similar pattern, and with peak shedding around day 2 or 3. So the bottom line is that these animals do shed a considerable amount of virus into their environment. This shows a little bit of the pathology. That panel A is basically showing infiltration of the subepithelium in the trachea with inflammatory cells and a bit of tracheal edema. Slides or panels B and C are the turbinates, so they get a again, kind of a mild to moderate inflammatory response in the turbinates. And of course, the turbinate epithelial cells is where all the virus shedding is occurring, I think, that is responsible for the oropharyngeal virus detection. That panel D basically just shows a section of lung that shows a mild inflammatory response, but we were unable to actually isolate virus from lung tissue. This shows the antibody responses over time. What you can see, as I indicated a minute ago, is that these cats develop just a super neutralizing antibody response following infection pretty routinely. The dogs, as I mentioned, they develop neutralizing antibody titers, kind of mild. And so I think what you'd have to say is that there has to be some virus replication going on in dogs. It's just not enough to readily detect virus in clinical samples. Okay, why is my, there we go. So here's the conclusion that we have reached in terms of dogs and cats. Dogs are minimally, if at all, susceptible. Don't throw them out on the street. They're really not, there's no indication that dogs are going to pose a risk of infection to people. And my interpretation of the literature is that the dogs that have, I think all of the dogs that have been shown to be PCR positive, for example, for SARS-2, are in households with COVID patients, and so they're undoubtedly just getting contaminated from their owners. Cats are a bit of another story. Similarly, if you look at the cats that have been reported to be positive in the real world, they all seem to be pretty much from households with infected humans. But they do shed a lot of virus. And so, I mean, it's certainly possible that an infected cat could infect a person. I think there's not much doubt about that. But I think the vast majority of cats that become infected are becoming infected from their living with infected owners. Happy to answer questions as we go along, or we can save them all to the end. That's pretty much the story on the dogs and cats. Now, as I indicated, we're kind of interested in host range, and you know, there has been a significant amount of interest on the part of livestock owners about whether COVID could infect livestock. And so we haven't done extensive trials, but we infected these three species. We've infected alpacas, again, by intranasal inoculation. We're unable to detect a hint of virus shedding. They did seroconvert mildly after infection. Goats we infected, oropharyngeal and nasal swabs on the goats were negative. We did again get low-level seroconversion. Cattle, we've done 3 baby calves, and they were pretty feisty and really didn't like nasal swabs, and so I really want to repeat this work. But we didn't detect nasal shedding of virus. So I think what we're planning on the cattle, I think it's still a bit of an open question. There's been some work in Germany, kind of similar to what we've done, showing minimal, if any, nasal shedding in cattle. But I think we probably need to do a little bit more work with cattle. Interestingly, we were able to isolate virus from the trachea in calves on 3 days after virus inoculation, so there's certainly some kind of replication going on. But in general, it seems like the ruminants are not a very susceptible group of species. Now, wildlife. There's a lot of concern about wildlife, particularly with respect to establishing reservoirs of COVID in wildlife species. I think that's a little bit of an overblown concern, but with the respect of the vast majority of virus transmission is occurring person to person. But we're interested in wildlife. We've done that with a lot of other pathogens, and so you can see some species depicted here that we have tested. There on the left-hand side are 3 species that do shed virus after inoculation. So deer mice shed a reasonable amount of virus. They also don't, and none of these species that we've tested develop significant clinical signs of disease. Or when I say significant, I mean really any clinical signs of disease. So Paramyscus deer mice, that second species is Neotoma wood rat. They also shed virus for a few days after inoculation. And we recently, in collaboration with National Wildlife Research Center personnel, we've tested skunks. And skunks are mustelids, kind of like ferrets, and I mean in the same kind of family, if you will. And they shed small amounts of virus over the first few days after infection. I haven't mentioned it yet, but one thing we know is that domestic laboratory mice are essentially not susceptible to COVID. And there was a question about whether wild house mice are infected, and the answer is no. We've inoculated a number of those, and we're not able to detect any virus shedding. We're in the throes of a large project not related to COVID with prairie dogs, and so we had the opportunity to inoculate a group of prairie dogs. They similarly did not shed virus after infection. Cottontail rabbits, a little bit surprising to me, but no virus shedding detected in cottontail rabbits. And then raccoons, of course. All of these would be considered what I would call peridomestic animals that could potentially interact with infected people. Raccoons, similarly to the other species on the right there, did not shed any virus over the first few days of inoculation. We're still working on the serology for those animals. Okay, let's see. Now, I thought I might just mention one kind of interesting situation. These are black-footed ferrets, and you may know that these are considered the most endangered species on the planet, really. The entire current population of black-footed ferrets was derived from 7 animals that were rescued a number of years ago, and we actually work quite a bit with the black-footed ferret restoration project. There's a breeding colony of black-footed ferrets north of Fort Collins, and when COVID first hit, they called me and they were extremely anxious about the possibility of their personnel infecting their black-footed ferret population and causing problems. And that's one of the reasons that we tested the prairie dogs, is to see whether they might be infected, because black-footed ferrets basically live off of prairie dogs. So we haven't done anything yet, but we have plans in the work to evaluate infection in a few of their retired breeders, and so hopefully we can get that going on in the relatively near future. One interesting aspect, as I mentioned, these black-footed ferrets went through a severe population bottleneck, and it turns out that they're all deficient for interleukin-6, which is a pro-inflammatory cytokine. And so my prediction is actually that they might be a little less susceptible than domestic ferrets in terms of COVID. But plague is going to be a lot bigger problem, is continuing to be the problem that just decimates these black-footed ferret populations. Now, I thought I'd just mention some other projects that we are, I'm really interested. We do a lot of conventional laboratory animal work in cages, but I'm really interested, especially in the real world. And so we have a number of studies going on with what I call artificial ecosystems. This actually is this is right after a room was cleaned. This is a room in our animal disease lab, a BSL-3 biocontainment building. And in this particular room, we've set up this artificial ecosystem to evaluate the transmission of monkeypox virus among species. So this is something that we started last year, and then it got basically just totally interrupted with all the COVID work. But what you can see is that we have a room with kind of some trees in it, some refuge boxes, common sources of feed and water. And into this room, we introduce what our plan is for COVID is to introduce some of these infected, not infected, these susceptible animals. So establish a population that has peromyscus, wood rats, hamsters, as I'll get to in a few minutes, species that are susceptible, and then introduce one or two infected deer mice, for example, and then just evaluate how that infection spreads throughout the population. So that's the plan that we hope to get going in the relatively near future. And now let me move on to these kind of conventional laboratory animals. As I mentioned a minute ago, and I think most of you know, conventional laboratory mice are not susceptible to SARS-2 infection. There's numerous people that just really don't want to do any animal models except mice. They don't have facilities, for example. And there was a couple of alternatives. There are transgenic mice that were actually created back in the SARS-1 day that express the human ACE2 SARS virus receptor. And there's a number of groups that have shown that those mice are indeed susceptible to SARS-2. Moreover, there's a couple of groups, Ralph Barrick and Barney in North Carolina and Barney Frank's group at NIH have engineered mouse-adapted SARS-2 virus that will infect conventional mice and induce a significant amount of pathology. And so there are people using those lab animal or those mouse models for SARS-2 virus. My own opinion is, and to be honest, I think a lot of people's opinion are that these mouse models are simply not all that robust. So we early on submitted a proposal to NIH to evaluate, to develop animal models, small animal models for SARS-2 infection. And NIH's call was to develop lethal animal models. And kind of the short story there is that that nobody has developed a lethal animal model for SARS-2. And so the species that we proposed evaluating initially were golden hamsters, ferrets, and guinea pigs. The hamsters and ferrets were, that choice was based on what people have done with SARS and also to a lesser extent influenza. And the guinea pigs were chosen just because they're used for a variety of respiratory diseases, especially tuberculosis. And so we've evaluated all three of those species, again, kind of standard intranasal inoculation of the animals with, we started off with 10 to the 5th plaque-forming units of virus and then monitor shedding, tissue burdens of virus, and histopathology. So let me just briefly tell you about ferrets. Ferrets are indeed susceptible to SARS-2 infection. They shed virus. They develop a mild pneumonia following infection, but they don't really. The amount of shedding and the pathology is not nearly what we found in hamsters. And again, several other groups around the world have found similar things with ferrets. And of course, ferrets are very closely related to mink, and I'm guessing you all know the story about the mink farms in the Netherlands. And so ferrets are a potential animal model that some people are using for SARS-2 infection. I think what I'm going to tell you is that ferrets don't seem to be as good of an animal model as hamsters, and of course they cost roughly 10 times what a hamster costs. And so I don't think we're going to see a lot of studies that utilize ferrets. They don't seem to have a big advantage over hamsters in terms of animal models for SARS-2. So let's move on to hamsters. We're doing hundreds of hamsters every week for a whole variety of most vaccine and therapeutic trials, and this is kind of the standard model that we've developed. We originally started with an inoculum of 10 to the 5th plaque-forming units, but we've done dose titrations, and we can readily infect hamsters with 100 plaque-forming units. And so for these therapeutic and vaccine studies, I've dropped the dose down to something a little bit lower, just so that we don't overwhelm the system with a high challenge dose. So our routine procedure is to collect oropharyngeal swabs on day 1, 2, and 3 post-challenge. And then we typically, for every treatment group, we have 2 cohorts, one that we euthanize on day 3 and one that we euthanize on day 7. On day 3, as I will show you on subsequent slides, we have very high virus burdens in turbinates, lungs, also in trachea, really minimal virus burdens in other extra respiratory tract tissues. On day 3, what we see histopathologically is moderately severe interstitial pneumonia. And so that's what we see on day 3, and I would call that the acute phase evaluation. On day 7, the virus has actually been cleared. So we've done quite a few hamsters and are not able to detect virus in turbinates and lungs on day 7. But interestingly, and I think relevant to the human situation, the pulmonary pathology is actually more severe on day 7 than it is on day 3, probably reflecting that so-called cytokine storm. So that's kind of the standard hamster model. Now, as far as clinical disease, I have up there largely subclinical with a couple of big question marks. And the big question marks relate to the fact that these animals' lungs are just trashed. As I'll show you subsequently, we're looking for some more. The reason I say subclinical is if you take these hamsters out of their cage and look at them, we don't really see any, overt reduction in activity if we have these animals, if we're evaluating them after pulling them out of the cage. But I just can't believe that they don't show reduced activity and that kind of thing. They do lose body weight, so we routinely measure them, weigh them daily, and they're losing 5 to 10% of their body weight. We're talking about control animals now, over that first week after infection. That bottom panel will give you an appreciation for the viral burdens on day 3 for tissues. This is from 42 recent control hamsters that weren't treated with anything. And so you look at turbinates, those are all in terms of 100 milligrams of tissue, and you can see turbinates 10 to the 7th or above. Both, we typically do 2 lobes of lungs, and those are typically in the 10 to the 6th plaque-forming units per 100 milligrams range. You can see the minimum and the maximum there. And then if you look at oral swabs, those continue to kind of surprise me. That 10 to the 4th range oral swab a day after infection just is a bit of a stunning figure to me, and that's clearly not residual virus. And we get a kind of a similar pattern on day 2, and then the virus titers in these oropharyngeal swabs tends to drop off relatively rapidly. We still see some virus on day 4 and a little bit occasionally on day 6, but it tapers off relatively rapidly. So high virus burdens on day 3, by day 7 it's gone. This shows you just a little bit of the pathology in these hamsters. So these are slides of trachea showing a abundant inflammatory response both within the tracheal tissue and in the lumen of tracheal glands on day 3, associated with quite a bit of virus. This shows a panel of the pulmonary pathology in a time course from on day 2, 4, 6, 14, and 28 days. And on the bottom, you can see the corresponding viral titers in lung. But if you look across that panel, we have significant inflammation even on day 2. It becomes very severe on day 6. Substantial histopathologic inflammation on day 14, but then it does start to clear on day 21. And so again, I think this kind of is similar to what people are thinking happens in humans. But the hamster do develop this severe bronchointerstitial pneumonia. These are just a few additional slides showing significant consolidation of lung. We actually have one of the pathologists here has a technique for measuring airspace over time, and they have a very significant reduction in alveolar airspace over the course of infection. This shows immunohistochemistry of the lung, and you can see abundant expression of viral antigens, both in that little bronchiole, in the epithelium, and then in alveolar epithelial cells, mostly type 2 pneumocytes. This is a little bit higher magnification showing bronchial epithelial cell replication of the virus and why we have abundant virus in those organ burdens. Now, let me. I'm happy to take any questions on this hamster model. We're using it. We just can't keep up, really. I'm spending a huge amount of time on all of these hamster studies. National Institutes of Health seems to be convinced that this is a very good model for evaluating countermeasures. We've actually had one of the fellows at a primate center saying that hamsters are probably a better model than non-human primates, and of course they're massively less expensive. So this is just an example of some data from the hamster model. We a number of months ago we've been involved with a technology for pathogen reduction in blood and plasma that utilizes UV illumination in the presence of riboflavin. And one of my colleagues, Ray Goodrich, had the brilliant idea that I wonder if it was working well to inactivate SARS CoV virus and other pathogens in blood. I might just mention that there's another interesting example. The transfusion world, of course, was another group of people that were massively concerned about the potential for transmitting SARS-CoV-2 in blood and plasma transfusion, even though there's really no viremia, but it remains a concern, and so they're interested in these pathogen reduction technologies. But anyway, Ray came up with the brilliant idea of why don't we just inactivate the virus using this Mirasol system and utilize that for a vaccine. And so we've done a substantial amount of work on that, and this so-called Solovax vaccine is actually being now funded by BARDA and NIH for further development. And so basically this just shows our initial hamster study using no vaccine, Solovax without adjuvant, and Solovax with two different adjuvants. And what you can see on that left-hand panel are the viral burdens in turbinates. These are basically oropharyngeal swab data. And what you can see is that the Solovax vaccine certainly induces or protects against turbinate infection, but certainly not completely. I actually think from these data and data from another number of different vaccines that it's vaccines, I think it's going to be not too difficult to protect against severe pulmonary disease, but I think it's going to be more difficult to protect against virus shedding in vaccinated people. The panel on the right-hand side basically shows virus loads in cranial lung. We actually evaluated these vaccines after both subcutaneous and intramuscular inoculation of the vaccine, and what you can see on the very right-hand side is 4 bars for control and the 3 different Solovax preparations. The control virus lung burden was about 10 to the 7th per gram, and we basically had essentially no virus in the lung on day 3 with the intramuscular inoculation. The subcutaneous inoculation worked less well. But that's an example of utilizing this hamster model to evaluate a vaccine. Now, I thought I would just mention to you other applications. We have a number of projects either in the works or about to be in the works where people want to nebulize drugs or antibodies into infected animals. And so this is just a poor man's way of nebulizing a group of hamsters. We basically just use one of those $20 Sprint nebulizers. This one contains an antibody, and we put hamsters in a jar and nebulize them within a biosafety cabinet. We'll see. We don't have much data yet on the efficacy of this treatment, but it potentially has human relevance. Another project that we're involved with is that again people are interested in is airborne transmission and how you might mitigate that, especially in a couple of different systems. This system basically was that I'm showing here was established as a prelude to some work to install ultraviolet UVC illuminators in ventilators, so with Philips Electronics. And so the idea is not just for SARS CoV-2, but for a variety of these respiratory diseases. It would be really nice if you could sterilize the air coming out of infected patients that are on a respirator. And so the way we set that up is to have 2 hamster cages. On the right-hand side, we have a cage with infected hamsters, and the air, these are ventilated, contained cages, and so the air coming in is unrestricted in that cage, but we have blocked the air outflow on that cage. There's a little tunnel that we've established between the 2 cages, and on the so-called recipient cage, we block the air inflow, but leave the air outflow intact. And so it's basically a way to see if air flowing from an infected hamster cage into an uninfected hamster cage can transmit the virus. And so we've done little of this, but the first trial that we did, the hamsters in the recipient cages were infected 24 hours after. We introduced them 24 hours after infection of the right-hand cage, and they were clearly shedding a lot of virus the following day. The mistake I made, or kind of the inefficiency of this system, is that we're going to actually have to do 1 recipient animal per cage, because hamsters also will readily transmit to one another. If you put an infected hamster in with a group of 3 other uninfected hamsters, the next day the whole cage is infected. So this is kind of the idea that I was mentioning for the ventilators, is that the next step in this project is to insert that UVC illuminator around this tubing and see if we can prevent infection of the recipient hamsters. This is what the setup looks like in our biosafety level 3 containment area. You can see the two different hamster cages with this tunnel, this tubing going between the cages. And what we are about to do as soon as I have a day free is to have this on one side, and then on the other side we'll have the UVC illuminator interrupting the airflow to a third cage. I'm destroying quite a few hamster cages, but Lab Animal Resources is fully supportive of this work. Okay, let me see what time it is. I've got a little bit of time left. Now, I mentioned early on that hopefully I've convinced you that these hamsters, if you just look at them, they really don't seem to be that distressed. But I can't believe, looking at the pulmonary virus burdens and especially the histopathology, that they don't have significant problems. So we're working on things like pulse oximetry to see if we can see what their oxygen deficit is. And then we also have recruited we've done this before for projects. We have the mechanical engineering students here, the seniors, have to do a little project in their senior year. And so we've recruited a group of senior mechanical engineers to add some brain power to our group and come up with a way that we can actually measure hamster activity. So back during the SARS-1 problem back in 2004, there was a group that basically installed these little hamster wheels into cages and showed a significant reduction in hamster activity by counting how much the hamsters ran around their wheels. I'm sure that makes sense. So they had hamster wheels that would count the number of revolutions and give you some data. And I think that's still a viable thing. The problem is that the cages that we use in BSL-3 for housing these hamsters are not tall enough to install those hamster wheels. And so that's been a little bit problematic. And also, I think we'd have to potentially have one hamster per cage also for that. So what we've got these mechanical engineering students doing is thinking about ways that we could put some kind of a mat on the floor that would actually be able to count steps, so to speak, very pressure-sensitive flooring, and get some index of hamster activity. These, of course, are mostly nocturnal animals. They sleep during the day. And then another idea that we just threw out to them is to have some kind of laser beam going across the cage, and we could count the number of times that beam was interrupted during the evening. And so I'm hoping that these young minds will be able to come up with a technique that is amenable, that is cost effective. The problem is that there's a number of techniques to monitor animal activity. Like there's some really spectacularly cool stuff done by a fellow at San Diego using cameras and fluorescent dots on mice, and they can track these animals around the cage and get a really wonderful appreciation for their activity. The problem is that the system costs about $3,000 a cage, and the work that we do, we're usually using like 30 cages per project, and so it's just not very feasible. We need something that's cost effective. And so that's what we've got these students working on. Bob, I think you were the one that gave me this picture, I think it's appropriate. I don't have anything else. I'm really happy to answer questions. I wanted to obviously I haven't done all of this work. We've got a big team here. And in fact, I'm noticing that I've left off some people embarrassingly here, but Angela, Erin, Stephanie, Isabella, Rachel, Paul, those are people that help on the day-to-day work with all of this. Jeff Root is the skunk raccoon expert. Amy, Lauren, Audrey are all veterinary students that we exploit, and hopefully they're learning quite a bit, and they're becoming expert at necropsying hamsters. Lindsay is the expert on the UV irradiation system. We've gotten funding from a variety of sources. And then, I clearly wanted to explicitly acknowledge how important it is to be partners with biosafety and animal care staff. And so Bob, of course, has ridden off into the sunset to some extent. But he was instrumental in getting us going on this. Heather Blair has been just wonderful in supporting the work that we're doing, and then the whole laboratory animal staff led by Keith Bruner has been wonderful in accommodating our needs for lab animal care. The people that I left off that embarrassingly are the pathologists, and so Sushan Han here at CSU has done. She's the one that provided those slides that I showed you early, and there's a PH student, Alex Bias, that also did the histopathology on the cats and the dogs. I'd be really happy to answer any questions, discuss anything with you. Thanks for your attention.
Well, thank you very much, Dick. That was just fascinating research. And we do have one question already in the chat room. That person said they're not familiar with the mink farm story. So if you could give a little bit on that, and then we'll see if we have other questions. Denmark, Netherlands, according to what I've read, and then I'll turn it over to you. According to what I've read, they produce most all of the mink that is used for garments, fur coats, those kinds of applications. But if you could tell them a little bit about those outbreaks and how it's been transmitted.
Yeah. So I can't remember I'm poor at timelines, but I think this started like 2 to 3 months ago. They recognized some mild respiratory disease in these mink farms, and as Bob said, it started in the Netherlands, and kind of the bottom line is that there have been some, I think there's reasonably, almost undoubtedly, these mink became infected. So they're very much like ferrets. They became infected by contact with infected people, but then they transmitted the virus among themselves. And I think there's reasonable evidence that mink actually then, through a reverse, through some zoonotic infection, infected people at these farms. And so there have been a number of these very large mink farms just totally depopulated. My own personal opinion is that I would be pretty excited if this was a way to eliminate mink farms from the planet. They're really, my take on this is that this is kind of a barbaric system that we really don't need. In fact, I read just recently that the Netherlands, kind of unrelated to COVID, I believe, have established a target of 2022 or something like that for banning mink farming. I think there's also been some reports there's also mink farms in this country, and I think there have been some reports of COVID infection in mink in Idaho. Maybe somebody else would remember that story better than I do. But it's that's another example of a reversed zoonosis where people infected the animals, and then in this particular case, pretty clear that the infected mink were a source of infection for some humans at least.
Yeah, that's essentially what I've read. And if people want to see numbers on that, you could go to ProMed, and they've carried that sometimes daily and definitely weekly, as Dick said, for the last 2 to 3 months. So I'm trying to get to some of the questions here, but I'm having a little bit of trouble seeing the questions. So Cindy, can you help me out with that?
I can read your questions for you.
Sure, that would be great.
So one of the questions is, has anyone come up with an estimated infectious dose in humans for comparison to animal models?
Yeah, excellent question. Somebody was just asking me that exact question yesterday. I have not seen that. As I indicated, we went down to 10 plaque-forming units, and we infected 1 out of 3 hamsters by intranasal inoculation. But I haven't seen those data for humans. You know, they are going to. I'm peripherally involved in a project to create a human challenge virus here at CSU that NIH wants, in case they actually decide to do human challenge studies to evaluate vaccine efficacy, and I really have not heard the discussion on what kind of challenge dose they're going to use for those studies, but I'm guessing it's, considering the rapidity of spread around the globe, I bet the infectious dose in humans is pretty low.
Okay. There was a question here about what happened to cat number 4. It was much higher than the others, but seems to be missing endpoint data.
Oh, those lines that you saw that stopped are animals that we euthanized to do the histopathology on.
Okay.
And so that particular cat was one of the, if I'm remembering correctly, it was one of the contact controls, and so it was infected a day or 2 later than the original cats, and so that's why the line was kind of delayed. And then we euthanized it to get all of the organ burdens and histopathology.
Okay.
And I can see one question here. It has to do with the animal models that you had. Do those animals shed virus via their fecal samples, and have you checked that?
Well, yes, that's another item that's been discussed quite a bit, and you know, we actually have done. I might just mention it. It's some of it's confidential, but we had just finished a project with another 40 cats for a vaccine study, and these were young kittens. It was a brutal study, as you can imagine, having to euthanize these friendly little kittens. But I think there's going to be demand for a cat vaccine. So bottom line is that these young kittens are far less susceptible to SARS-2 in terms of both, especially in terms of the amount of virus that they shed. So in that particular study, we did do fecal shedding and rectal swabs, and we really didn't find any virus. So I think, you know, there are a number of there's a number of other potential complications of COVID infection that I'm sure everybody's read about. So gastrointestinal disease, cardiovascular disease, and they, you know, I think they're going to be studied, but they don't seem to be prominent sources of neurologic disease. That's another one. I mean, I didn't mention it, but in these hamsters, we pretty routinely see a low-grade meningitis. So the virus is in the brain, no doubt about it. And you're probably familiar with the loss of taste and smell that's experienced by COVID patients. So yeah, I think GI infection probably occurs. It's the vast majority of that work has been done with PCR, and so it's hard to know how much of that is just viral RNA. That's my speculation, is that there's not much replication in the GI tract. It's all viral RNA from ingested virus from the turbinates.
Okay, couple more questions for you. Have you thought about using the piezo sleep machine that monitors sleep disruption patterns for the hamsters? Or looking at the behavioral patterns that might differ from the infected and non-infected individuals?
Well, that's exactly what we want to do. But to be honest, I'm writing that down. Spell that for me. I don't know about this sleep machine.
P-I-E-Z-O, sleep.
Okay, I'll have to look that up. You know, I actually bought a couple cheapo Fitbits watches and tried to duct tape them to hamsters, and it just didn't work at all. But I'll have to look this one up. I'm not familiar with that.
Okay. And just a comment here, it was made by one of our attendees that said, I just want to make a comment that Dick Bowen's research epitomizes the extraordinary need for researchers, clinicians, engineers, biosafety, wildlife biologists, vets, etc., to share the same sandbox when solving these problems.
Well, I completely agree. We collaborate with a whole bunch of people, and as kind of intimated there, each one of those little worlds can be a little bit insulated sometimes, but it's really great to get a comprehensive picture.
And then the last question I see right now is, have you seen any age-related differences in your hamsters? Are you working on any longer-term recovery studies?
Yeah, that's kind of like two questions. That's kind of on our list of things to do, to look formally at age-related differences. We haven't done that yet. I think you can buy retired breeder hamsters, and so that's on our list for something to do. And then also to look at little kind of very young hamsters. So I think that's a really important thing to do. The hamsters that we use are typically about 8 weeks of age. So that's important. Now, the other thing I haven't mentioned, it was actually, we've done this on a couple of different studies vaccine studies where one of the vaccinations, so to speak, was infection with SARS CoV-2. And so the kind of bottom line there is that if we infect hamsters with SARS CoV-2, let them recover, and then re-challenge them at 8 weeks after their primary infection, they are very well protected. But even then, there's a little bit of breakthrough in terms of virus recovered from turbinates. We don't really know if that's input virus or replication. We need to do some immunohistochemistry on that.
Okay. And just back on the piezo piece, the person who had suggested that piece of equipment says they use it with mice.
Really?
It uses a pad under the cage to monitor activity or movement that demonstrates sleep bouts and activity.
Wow, excellent. I'm so grateful for that information. I'll be looking that up as soon as I sign off here.
Okay, and that's all the questions I'm seeing at this point in time. So at this point, I think we'll turn it over to Denise.
Well, let me just thank Dick for this excellent presentation, and it's typical of the research that he and his group of researchers do. Dick always has veterinary students involved in his research, as well as graduate students, postdocs, collaborates with just a host of people, kind of that list that came up there, engineers even, and gets everybody involved in it. It's just always fun to work with him and collaborate with him on his research enterprise. So thank you again, Dick, for this.